In part 3 of Guinea Pigs, Humans and Carrots, I mentioned the protein data bank, which is a repository online of three-dimensional information about molecules like proteins and nucleic acids. It's quite easy to find. Just search for protein data bank, and the first link will be to www.rcsb.org. So we'll open that up, and this takes us to our main page after a moment. So there's lots of information here about the three-dimensional structure of proteins. One thing I noticed here when I was looking recently was this competition they ran with American high schools, asking high school students to develop three-dimensional models of proteins based on information in the data bank, and these were the winners from South Brunswick High School with their model of a capsase protein. Capsase proteins are proteins that are involved in apoptosis, that is to say, programmed cell death. So that looks quite a nice thing to do. We'll have to find a way to think about doing that in the future. Okay, we'll go to the main page. I've got the search option. We're going to search for collagen. This molecule we're interested in, and immediately finds it and offers a number of things. One thing you'll find is lots of enzymes which are involved in collagen, but what we're interested in is collagen itself. So I'm going to go to the molecule of the month. They have a molecule of the month facility, like many of these databases. And it comes up with a couple of structures which we'll talk about in a bit. This is why I've gone to the molecule of the month facility, because it goes into a little bit of detail. So there's quite a lot of stuff about collagen there, including some images you will recognise from the videos which I've done, and a couple of structures there: an idealised collagen and a human collagen. We'll have a click on the idealised one, and after a moment it'll open up its three-dimensional structure. This is run in what's called JMOL, which means that you will need Java installed on your computer. Now, if we click on the image, you can do all sorts of things. You can rotate it, you can make it a bit bigger, a bit smaller by using the mouse wheel. If you look down for the information here, we'll see at the moment it's called what's called cartoon structures, and that's actually what these things are called. We can have different ones. We can have one which is providing the backbone information, so you can probably see the 3D helical structure perhaps a little bit more clearly there than you could in the cartoon structure. It has the CPK structure, which is the conventional way of colouring atoms in models. There's not many different ones here, but when we do the molecular models tutorial a bit later on, you'll see more. And here's an example of a molecular model. The colours in this case, a blue is nitrogen, a red is oxygen, the white's are hydrogen, and the black is carbon. Obviously atoms don't have colours because they're smaller than the wavelength of light, but conventionally when we draw molecules or we create models, this is how we colour them. Okay, so as I say, there's a number of things we can do here. I'm going to go back to the cartoon structure. I'm going to have a little bit of a look at other ways in which we can look at it. We're looking at it by chain, which is immediately coloured in the chains differently. So we can zoom in and have a look at these, and we can see the three different chains, and you can manipulate this, and with a little bit of three-dimensional imagination you can see the left-handed helices that make up the overall right-handed helix of collagen. Another thing we can do is look at it by hydrophobicity, so picking out those groups which are hydrophobic and those groups which are hydrophilic. And the easy way to see which one is which is to look at it end on. Zoom in there a little bit. As you might imagine, the sort of red groups are on the outside are those which are hydrophilic, whereas the hydrophobic groups, in this case glycine, are on the inside, tucked away and protected. And if you click on it, it comes with a little bit, it says the proline residue there, hydroxyproline residue there. I can't find a glycine one at the moment, but there is one there. There's a glycine one there, and hydroxyproline there. So it's quite useful. It gives you some indication of the three-dimensional structure. In this case, clearly describes one of the key elements of protein folding, which is hydrophobic groups are tucked away on the inside, whereas hydrophilic groups are on the outside, exposed to water. Just while we're here, we'll just have a look at the human collagen one, which is an actual collagen. It'll fire up in a minute. The diagram is a little bit different, and in some respects it's a little bit more clear, so we'll zoom right in here. We can again easily see the three chains there. perhaps a little bit more easily than we could before. Which, as I say, if you look carefully, you'll see a left-handed helices, whereas the overall thing is a right-handed helix. Again, we can pick out hydrophobicity, perhaps a little bit less clear. I don't know, that's not bad, is it? So you can see the hydrophobic groups tucked away on the inside, surrounded by the hydrophilic groups. That's the protein data bank. Lots of useful information on here if you're interested in proteins and the three-dimensional molecules. So have an explore of it yourself. Okay, thanks for listening.